Hi guys, and welcome to Biological Sciences for Year 10. This is activity 6A, which is going to look at single cross problems. Now, a single cross problem is basically what happens when you get one trait which joins with another trait, and we work out the probability of what the offspring are likely to get. Now, at no point are we going to say they're definitely going to get this, because in each case you've got two different sets of gametes with two different, or often it might be two different types of information, so you're never too sure which half of the gametes are going to join together from each parent. Now what I've done is I've set up there's three examples in here. I've set up a basic slide to run you through, and then I've also given a slide where I've done a cross with the Gene Screen app so that you can use either to determine what your genetic crosses are. And then you'll also find that in this section of the course there's another activity called 6C. 6C will run through some harder sort of questions looking at how do you determine genetic genotype from ratios, or how do you deal with pedigrees. So you actually need to watch both 6A and 6C to help you with 6B. 6B will run through some easy questions, and then they will get gradually harder as you get down the bottom, and then obviously we'll put some in the pop quiz at the end so that you can really have a good go at these genetic crosses. Okay, so the best way to actually deal with these single crosses is to run through a cross first and take it from there. So here we've got a situation where we've got sweet peas and we've been given a yellow seed which is dominant over a green seed. And we've got to predict what happens to the offspring if we mate a homozygous yellow-seeded plant with a green-seeded plant. Well, homozygous yellow means that yellow, so they're they're exactly the same genes, so we need to have a code for what's the yellow, and obviously the green seed is going to be homozygous recessive, so we're going to have a code or a letter which is going to denote the recessive type of the gene. So this is how you would go through it. First thing you've got to do is identify the alleles. What letters are you going to use to correspond to yellow, dominant, or the recessive type? Now remember, they should be the same letters. One is going to be a capital, one is going to be a lowercase, and when you write them they should be clear so that you can see what's going on. Then write down the phenotype from the question. Phenotype basically is what is the physical property of the parent, and then write their genotype, which is what is their genetic properties in the parent, and that will determine whether it's a homozygous or heterozygous. So you'll be having two letters there. If they're homozygous, obviously those two letters are going to be the same. If they're heterozygous, then the two letters are going to be different. Then you make a Punnett square, and you basically separate into gametes, and so you'll have two gametes for each parent depending on the type of organism or genotype that you've got there. And then all you do is then you say, well, what happens if one gamete meets another gamete? What would you get? And you'll get 4 possibilities, and from those 4 possibilities you can then make a probability statement as to what the offspring are likely to be. So let's run through it. So the first one, we're going to determine the alleles. Now in this case yellow is going to be dominant, so I'm going to use this capital Y to determine the yellow. Green is going to be recessive, so a lowercase y will determine a green seeded plant. So we've got capital Y and little y. Now our parents, we know, have a phenotype of one's going to be yellow and one's going to be green. Now we've been told in the question that one of the parents is going to be homozygous yellow. Now homozygous means the same, so it's going to be capital Y, capital Y. We've been told that the green is going to be recessive, so it's now going to be homozygous recessive, so it's going to have lowercase letters, so it's little y, little y. I then bring up my Punnett square and I split each of the capital Y's and each of the lowercase y's into gametes, into half, because remember gametes divide by meiosis, so they have half the number of genetic material, so that when they come together you get a full complement of genes. So let's split them up. There's capital Y, and we've got little y, little y. So then we say what happens, we start in the left hand corner of our square, and basically we say what happens if each of the gametes meet. So in this case we get capital Y, or capital Y, capital Y, capital Y, capital Y, little y, or capital Y, little y. Okay, so you can see here we've got 4 different possible outcomes. Now in this case the 4 different outcomes are that all of the offspring are going to be heterozygous yellow. Okay, so you'll get 100%. Doesn't matter how often you cross them, they will always be heterozygous yellow. Okay, so let's look at a second example. Now in this case, what we're going to do is we're going to cross a homozygous yellow-seeded plant which is crossed with a heterozygous yellow-seeded plant. So we follow through our process again. What are the alleles? Well, we're going to use our yellow, our capital Y, little y again. So we've got yellow, yellow, that's the phenotype. They're both going to be yellow, but one's going to be homozygous, which means they're the same letter, and one's going to be heterozygous, which means they're going to be different letters. So you can see here we've got capital Y crossed with capital Y, little y. So we now pull in our Punnett square and split them up into gametes. So there's the first set of gametes, that's the homozygous yellow, and there's the second set of gametes, now that's the heterozygous yellow. And then we do our crosses. So we've got capital Y, then we've got capital Y, little y, capital Y, capital Y, capital Y, little y. Now there's no difference in the actual phenotype of the parents. However, out of them we're going to get 50% will be homozygous yellow and 50% will be heterozygous yellow. Okay, so that basically it's exactly the same as the previous one, but you've got a different probability. Now if you were then to cross those offspring, you would then get another situation which we'll look at in example 3. But first let's run through how we could use the gene screen app to do the same sort of thing. Now the first thing we're going to do, you can see here we've got homozygous yellow seeded is crossed with heterozygous yellow seeded. So the first thing we've got to do is go to our gene screen app, and this is our front page. So you press on the icon which basically asks you to do the blank Punnett square on the front screen. And then you'll get a screen that looks like this. So what you then need to do is to press on one of the letters down the side and choose the letter which corresponds to the trait that you want to show in your genetic cross. So in this case we're going to pick Y. So I tap on Y and this is what I will pull up. And you can see here that we've got the homozygous dominant, we've got the heterozygous, or what they call the carrier, and then we've got the recessive type, your homozygous recessive, which they're calling the affected, and this is because they're talking about diseases, but it still works for what we want to do. So you've got your 3 different types of genotypes up the top there. So your next job is basically to pull down your desired areas. Now it doesn't matter which way you pull them, but because you'll still end up with the same type, but we're going to have our homozygous dominant, our capital Y, and our heterozygous, which is going to be capital Y, little y. So we pull it down, each one, so you just drag onto it and it says drop here. So you just click onto that button and you can see down the bottom that you've got your capital Y, and your capital Y, little y. Okay, so the next thing you've then got to do is hit the bottom tab which says mate. And what will happen is you'll pull up this slide. Now this slide will then show you all the various genotypes straight away. So you can determine just exactly as we did earlier, you can see that we've got our 50% homozygous dominant and our 50% heterozygous, and they're all yellow. So basically that will allow you to use your gene screen map. So when you go through your examples, all you have to do is put in your buttons, put in your alleles, hit the buttons, and then you'll get your probability. It'll save you a wee bit of time. However, remember in the test you'll be expected to write it, so probably practice a bit of both and you should be fine. So here's our third example that we're going to run through, and in this case we're going to have a heterozygous yellow seeded plant joined with a homozygous green seeded plant. Remember in the previous examples where we've had homozygous, they've always come out yellow. Then we've had homozygous heterozygous, still come out yellow. Now we're going to have a situation where we're going to be using our green seeded plant joined with a heterozygous. What's going to happen? So there's our process again, making sure that you understand the process and go through it bit by bit. Once you've learnt this, you won't need this process anymore, you'll automatically do it. So what are our alleles? We've already determined those. We're going to use capital Y, little y. What are our phenotypes? So we've got yellow and green. What are our genotypes? Well, we know that we've got heterozygous yellow, so it's going to be capital Y, little y, and we've got green, which is homozygous recessive, and then we place it into our Punnett square. So there's our yellow heterozygous, and here's our homozygous recessive, and now we cross them. Capital Y, little y, capital Y, little y, little y, little y, little y, little y. Now straight away you can see here our homozygous recessive is coming through. So what happens if we have a heterozygous plant meeting with a homozygous recessive plant, and what we end up with is 50% are going to be heterozygous yellow and 50% are going to be homozygous green or homozygous recessive. So this basically deals with all the various types of genetic crosses that you can have for a single cross. And if you follow these 3 examples throughout all of the exercises, you won't go wrong. You just follow through logically, writing down what you've got in each case and then stating what happens at the end. Remember, each set of mating will always have that same cross. So if I was always to cross a heterozygous yellow with a homozygous green, their first mating would be 50% heterozygous yellow, 50% homozygous green. The second mating of that same ratio would be the same. And that's important to know. It doesn't change for each mating. So you need to bear that in mind. Okay, so what I'd like you to do is now have a go at exercise activity 6b. And yeah, good luck. If you get stuck, by all means come back and watch the video. And don't forget, watching activity 6c might be a good idea for some of the harder problems, but I didn't want to confuse you. The first couple of questions that you've got in the activity will run through those 3 exercises. So what I suggest you do is probably do the first 2 and then go and have a look at activity 6c. Alright, thank you for watching me, and join me again for our next activity.